Hi, my name is Vaughn Cooper, and I'm a professor at the University of Pittsburgh in the School of Medicine. I'm an evolutionary biologist and a microbiologist, and a co-founder of our Center for Evolutionary Biology and Medicine. I'm so proud to share with you a program that we think is the most significant part of our work in research and education. The overall mission of our lab, shown here in a recent distance picture, is to share the wonder and the power of tracking evolution in action with everyone. We do this in three major ways. First, my research lab at Pitt is working to understand fundamental processes in microbial evolution, such as how bacteria adapt to grow on surfaces or host organisms like us by producing biofilm, or how they evolve to resist antibiotics, and how we might predict these evolutionary paths to help medicine. Second, to study evolution in action, we've developed methods for high throughput DNA sequencing and rapidly analyzing these data to identify the mutations that cause new adaptations. Over the past 5 years, we've shared this capacity with hundreds of research labs, and more recently, we've spun the center off as a company, the Microbial Genome Sequencing Center, or MIGS. We're especially proud of the many small colleges who use MIGS to advance their research and student learning. But this video focuses on the third and hopefully the most far-reaching way we're sharing the wonder of learning evolution as it happens. This is our research and education program called EvolvingSTEM. EvolvingSTEM aspires to become a comprehensive life science education program that's built on authentic research from our laboratory and many collaborators. It's been under development for 8 years, and I've been pursuing this dream behind EvolvingSTEM for 20 years. In this curriculum, students conduct a real experiment where they witness evolution in a test tube within a week, and then they analyze the whole genomes of bacterial mutants isolated by other students using pro-level bioinformatic tools.
So what we love about evolution experiments is their ease of access and the inherent excitement of discovery. The Pixar movie Ratatouille is about a mouse that dreams of becoming a chef, and early on, he learns an important lesson from his idol, Chef Gaston.
Who is that?
Oh, him? He's nobody.
Not nobody. He is part of the kitchen.
Yeah, he's a plongeur or something. He washes dishes or takes out the garbage. He doesn't cook.
But he could.
No.
How do you know? What do I always say? Anyone can cook.
Well, yeah, anyone can. That doesn't mean that anyone should.
Well, that is not stopping him. See? What is he doing?
No, this is terrible. He's ruining the soup, and nobody's noticing it. It's your restaurant. Do something.
What can I do? I am a figment of your imagination.
But he's ruining the soup. You gotta tell someone this.
So the point of that snippet was to show what Gaston, the chef, championed in that Pixar movie, Ratatouille, is the idea that no matter how ambitious the recipe, anyone can learn to cook and find reward from the process. We think that in much the very same way, anyone can learn how evolution by natural selection acts on genetic variation to produce adaptations. In fact, in identifying the root cause of new adaptations remains a grail of evolutionary biology. Wouldn't it be amazing if any student could share this excitement and experience the wonder of being the first person to discover a new mutation responsible for an adaptation? EvolvingSTEM is based on an experimental method that allows teachers and students alike to explore how populations of microbes change in real time to become fitter in new environments, including on man-made surfaces, during infections, and in response to antibiotics. So first, I've got to share some history and scientific background about how we got here. Back in 2007, my laboratory was thinking about ways to better understand how microbes evolve in biofilms, because even though microbes live mostly on surfaces, in aggregates or in films, much of our understanding of their evolution was based on experiments in well-mixed liquid cultures. And so what we soon realized is that biofilm is not just a binary switch from swimming to sticking, but rather it's a life cycle of attachment, biofilm production, dispersal, and reattachment. We took the simple approach of adding a plastic bead to a test tube containing growth media, and by moving this bead from a new test tube containing a fresh bead each day, we select for bacterial mutants that attach better to this bead, but also that can disperse and reattach to colonize the new bead each day. In fact, our simple bead model replicates this life cycle every day. So over the following years, we worked to miniaturize this so it can be delivered as an evolution kit for classrooms. First with large disposable glass tubes, and now with smaller disposable tubes. The requirements of our kit are now minimal. The experiment can be conducted almost anywhere, and the kit itself fits in a box. What's really exciting is we learned soon by doing research with this model that with the opportunistic pathogen bacteria, Burkholderia senescipacea and Pseudomonas aeruginosa, both of which are good at forming biofilm, that they rapidly diversify in a few passages in our bead model and produce different colony morphologies. And when we pick these colony morphologies and regrow them, they keep their morphology or they breed true, meaning that they're heritable mutants. In populations of both species, some types are especially good at sticking but not dispersing, so they remain rare, and others that balance these strategies are more common.
Our next exciting discovery was that when these different mutants grow together, they produce more cells and biofilm together than they do when they're growing by themselves. They produce synergistic communities as they assemble together in biofilms, and in fact, colonized distinct niches within the biofilm. So when I saw these bacteria evolving and diversifying in real time, producing different niche specialists, I thought, this is how everybody ought to experience evolution, by doing an experiment with microbes evolving in real time. We wanted to share this experience with students for all ages, and this became the catalyst for EvolvingSTEM.
We began to work with the safe bacterium Pseudomonas fluorescens, a plant probiotic that's related to the pathogen Pseudomonas aeruginosa. It grows well at room temperature, and it's good at forming biofilm. It's also fluorescent under UV light. Here you can see an individual cell fluorescence, and this is one of their biofilms. Students start the experiment on a Monday, and after transfers on Tuesday, Wednesday, and Thursday, they plate their evolved cultures on Petri plates on Friday and look to see if the round colony types in their starting cultures have changes. This selects on randomly arising mutants in the cultures and enriches those that stick to the bead better. In this illustration, these are the yellow bacteria that become more common or more frequent over time. Students can see the tubes producing more biofilm over time at the air-liquid interface as slime. And when they plate these bacteria by diluting them and spreading them on agar, they often produce round, different looking colonies from the uniform round colonies produced by the ancestral strain of Pseudomonas fluorescens. So just over a few days, the population has diversified. Further, the cultures grown by different student groups often have varied results because the process of evolution by natural selection depends upon new mutations arising at random. Different mutations equal different adaptations. Now, I must acknowledge the amazing researchers and teachers who have helped make EvolvingSTEM the full-fledged curriculum that it is, that grew out of these experiments. First of them is Dr. Abigail Matella, who's currently leading and building this program, Taylor McLeod, who really helped found it, super teachers in New Hampshire who took the first plunge, Mike Handwork, Shani Scarponi, who continue to offer it in New Hampshire at Winnicott High School, Drs. Ron Kinzer and Dr. Edwin Kitchington, who've really innovated this program here in the city of Pittsburgh. Rebecca Brewer, who was the first to adopt this program fully remotely in Michigan, and also not pictured, the first teachers to take the plunge here in Pittsburgh, Brendan Albright and Jacob Warren at Peters Township High School, and several other teachers listed here who have been really critical in fleshing out our program and giving it their own personality. I want to emphasize that these teachers really have come to love it, and you can too, by requesting elements of our program for free at our website, evolvingstem.org. Over the last 8 years, more than 2,000 high school students have experienced our program at 11 high schools. In addition, 7 colleges and universities are adopting our program, and there's a lot of opportunity there. They're in 4 states and 6 states respectively. For students in high school, we've documented a 50% increase in performance on subject matter assessments and significant increases in student motivation towards STEM, especially among women. It's also important to emphasize how many new skills students and teachers alike can learn through this program. Students learn biotech skills, including pipetting, dilution, sterile technique, foundations of microbiology, experimental design, controls, replication, and simple statistics. But wait, there's more. There literally has never been a better time for all of us to learn how to decode microbial genome sequences. This is so relevant when we're investing in tracking the novel coronavirus genome, for instance, by sequencing its genome. So we can ask, do the evolved mutants that the students are isolating from their test tubes differ genetically from their ancestors, and how can we find out? So how do we find the genetic causes of these different types? We use the same methods that we use in a research laboratory, for example, to track SARS-CoV-2, to identify how bacteria become antibiotic resistant, or to identify these new mutations. We can pick one of these evolved clones with a new heritable colony type, isolate its DNA, and then completely sequence it quickly using next generation software. This is the goal of much of our research, to define and understand the genetic pathway to an adaptation. And it's also the key learning experience of our bioinformatics curriculum. We're very excited about this curriculum because, first, it's ideal for computer-based research in this remote or restricted learning environment of COVID. Second, because bioinformatics is one of the fastest growing careers, which blends computer science and biology, and skills that are learned in these tracks are broadly transferable. And third, students learn about how mutations produce new phenotypes, the core of modern genetics. So I'm not going to go through the details of the bioinformatics curriculum here today, but I want to provide a link to another YouTube video that shows how the program works in greater detail. In this program, students will log into a website that's dedicated to understanding understanding the genomes of these microbes, and especially cause of disease. Students upload the data to their chosen mutant, compare the sequences of their mutant with the genome of the ancestral strain, and then they use powerful software to identify the mismatches between the genes, their sequences, and the reference genome. They then identify the affected gene. They then look up that gene at a database about Pseudomonas and ask, what does this gene do? It turns out that many of the mutants producing wrinkly colony morphologies alter a network of genes that help bacteria sort of decide whether to stick or swim by producing biofilm or activating their propeller-like flagella. Some of these are in the so-called WISP, or wrinkly spreader pathway. The way these mutations produce adaptations is by increasing concentrations of a signaling molecule that governs this stick or swim decision. This molecule is known as Cyclic-di-GMP. So some affect the WISP pathway, some affect this YFI pathway, and some affect this MORT-A pathway. These mutations that increase concentrations of this molecule wind up increasing biofilm production, and this is an adaptation. This is the central governor of the cell fate decision. Dispersal, in contrast, would be triggered by low Cyclic-di-GMP. So how are these mutations relevant to our broader understanding? It turns out that these genes are also under selection when the pathogen Pseudomonas aeruginosa colonizes wounds to cause infections. Pseudomonas aeruginosa is a related species that's a major opportunistic pathogen. It infects persons with cystic fibrosis, patients on ventilators, burn victims, and other people who are immune compromised. The WISP pathway found in Pseudomonas aeruginosa and its related machinery are nearly identical in Pseudomonas aeruginosa, and the very safe Pseudomonas fluorescens. And further, wrinkly colony morphology mutants often evolve during Pseudomonas aeruginosa infections. So it turns out we find mutations in exactly the same signaling systems becoming detectable within days in experimental models of burn infections. And the biofilm phenotypes that result are associated with greater antibiotic resistance, increased inflammation, and delayed wound healing. We sequenced the genomes of Pseudomonas aeruginosa isolates from these model wound infections, and these mutations that cause the high biofilm phenotype exclusively in the genes WISP-A and WISP-F. The wrinkly mutants produced these WISP mutants produced these wrinkly colony morphologies. And when we replaced or complemented the mutant genes back with the ancestral WISP-A allele, the colonies became smooth again, showing that it was just this mutation in this WISP gene that was responsible for the wrinkly colony morphology. So again, the wrinkly colony morphology evolving during the wound infection of Pseudomonas aeruginosa is caused by WISP, just as the wrinkly colony morphology that produces the wrinkly colon produced by Pseudomonas fluorescens is also caused by mutations in these genes. I hope I've shared our excitement about this EvolvingSTEM program and its ability to teach students how natural selection can quickly act on newly arising mutations to enable microbes to adapt to growth on surfaces or in infections. Students learn key methods for a career in biotechnology and analyze bacterial genomic data isolated from student experiments to identify the genes that produce these adaptations. A student who completes these programs will gain key skills required for advanced microbiology or genetics research in a wet bench laboratory, and also develop a foundation for further work in bioinformatics. Students will also achieve greater proficiency in a range of next generation science standards. Thanks for watching. For more information about these programs, please visit our website, evolvingstem.org, where you can browse the available curriculum materials or submit an inquiry for free or low-cost materials for your classroom or laboratory. We look forward to hearing from you, and thanks for watching.